My name is Jason Walker, and I am a biology coordinator with the National Math and Science Initiative. And this screencast is going to be about viruses. Now, this is in our biotechnology unit. Okay, so just tell me what you see here. Okay, so basically, what we have here is just a bacterium that is being attacked by bacteriophages. It's being attacked by viruses. Okay, so what this kind of helps us understand is just the sheer size of these viruses. I mean, we know how small bacteria are, but look at these viruses, right? So these are bacteriophages. They are viruses that attack prokaryotes. Okay, and since these virus particles are attacking the bacterium, I want you to think about: are they feeding? What are they doing here? Do you think that they're feeding? Do you think that this is some sort of part of a reproductive event? Or what does this look like to you? Okay? Do you think these viruses are alive by looking at this picture? Okay? So I just want you to keep thinking about these things as we progress through. Okay? So let's talk first about what actually is a virus. Okay? So in the late 1800s, Martinus Beernik performed this experiment. Okay, basically what he did is he extracted some sap from a tomato plant that he knew that had this disease called tobacco mosaic disease. Okay, and he passed that sap through a filter. Okay, and that filter filtered out prokaryotes. It was small enough, okay, that it could stop all the bacteria. So whatever got through had to have been smaller than bacteria. Okay. So and then he rubbed that sap on leaves of a healthy plant and noticed that that plant then became infected. Okay. So that's the result of the experiment. The filter sap could in fact infect healthy plants. So we continued to replicate this experiment on previously healthy plants which had become infected due to sap, and was able to show that the infection rate did not decrease. Okay? So in 1935, Wendell Stanley actually was able to crystallize this infectious particle for tobacco mosaic disease, and it was a virus, something much smaller as we saw in the previous picture here. Okay? And it's been since known to be what's called tobacco mosaic virus. Okay? So let's look at these types of virus here. Okay? We've got some that are cylindrical shape. We have some that look like little spheres. One over here looks like it's a little tiny little spider. Okay, so lots of different shapes, but what are the commonalities? What are some similarities that you see between these? Okay, what do they all have? What do some of them have? What some of them do not have? Let's look at these. What do they all have? Well, they all have some sort of capsid. Okay, even though the capsid on the bacteriophage is much more complex than the others, they all have some sort of capsid. Okay. And they all have, what else? Genetic material. Now, maybe that's DNA, maybe it's RNA, but it's all about this proteinaceous capsule or covering that is surrounding genetic material, DNA or RNA. That's basically what a virus is. And here in a little bit, we're going to talk about protein synthesis and how our cells actually undergo transcription and translation. And they're making viruses. So let's think about this for a second. What is transcription translation making? What makes protein? What is a virus? It's just protein that encapsulates genetic material, right? Let's look at these just for a little bit longer, and let's think, how are they different? How are these four different main types different? Well, some have a membranous envelope protecting that capsid. Okay, you can see this membranous envelope around these capsids inside. Okay, remember the capsids are surrounding what we're calling our genetic material, DNA or RNA. Okay? Some of them have glycoproteins sticking out from the membrane. Now, we have membranes around our cells, and we have these glycoproteins, and this is going to become important to remember here in just a little bit when we start talking about how in the world are these viruses actually made, right? So, what do you think that these glycoproteins are for? Okay, why would a virus want to have a membrane around it, and why would they want to have glycoproteins? Well, what are glycoproteins used for on our cells or on other eukaryotic cells? Just keep that in mind. And they all have different shapes. Third thing is they all have different shapes. Okay? So now let's think about when in biology, when we talk about a specific cell or a specific species, an organism's structure. If you were to change the structure, what do you also change? Well, the function, right? Change the structure, you change the function. So you can actually think all of these virus particles have different shapes. So they probably have what? Different functions. Let's look at just real close at the components of a virus. Okay, so what you see here is it basically consists of protein and nucleic acids. Those are the other two. These are the two main biological molecules that we're talking about here. Okay, basically there's a protein capsid that's encasing this genetic material. Okay. The capsid is actually made up of what we would call, and many times what we call capsomeres. Okay? But again, you can also see there's some accessory parts to these. Now, what other biological molecules do you think make up these accessory structures? Okay? Things like when we come back here and we think about these glycoproteins, what does glyco mean? Where do we see things like words like glycolysis? What does glyco mean? Well, glyco means sweet. Right? Glycolysis is the process by which glucose or a carbohydrate is split in the very first part of cellular respiration, the very first step of cellular respiration. Right? You give off the 2 ATP, you split something sweet, glycolysis, sweet splitting. Right? And so if we have glycoproteins, and we have phospholipids because you see this has a membranous envelope on there. We have a plasma membrane as well made up of phospholipids. So we know that there's proteins, we know that there's lipids, we know because those phospholipids have those fatty acid tails, right? Well, they also have, so not only do they have the proteins and the nucleic acids, and not only do they have glyco, they have the carbohydrates, but they also have the lipids, right? So they have all four main biological molecules right there. Okay? Now, there's always let's talk about this just real fast. There's always genetic material that's involved, DNA or RNA. Okay, that is a nucleic acid. Okay, now it could be DNA. I want to stress, it could be DNA and it could be RNA. The RNA could be single or double-stranded. The DNA could be single or double-stranded. And I know that in your class you've learned since they started teaching you biology, DNA is double-stranded, RNA is single-stranded. But there are instances, especially when we talk about these viruses, where that doesn't necessarily hold true. So how would we know whether a virus has DNA or RNA? Well, if we were able to look at the code, we would be able to see that even if DNA is single-stranded, it still has thymine, right? And RNA may be double-stranded, but it still has uracil. So that would be a really great way to figure that out, okay? So we said the genetic material is encased in a protein coat with other protein accessory structures, and it has the lipids, it's got the carbohydrates, and so let's talk about the basic reproduction of a virus. This is basically what it is, okay? So you have the virus will inject either itself or inject its DNA inside of a host cell. So here's the host cell. Let's just say that this is our cell. Okay? So it injects itself or its genetic material into the host cell. The genetic material then replicates and or is transcribed into viral RNA. So here's the viral DNA that came from here. Okay? Here are the little capsid proteins that came in. So here's the viral DNA. So basically what happens is that our DNA gets shut down. The viral DNA will just shut our DNA down. And then through transcription, our cell will undergo transcription and will use the viral DNA to make viral RNA. So what is that viral RNA then able to do? Well, it's able to make more what? Through translation, it's able to make more protein. So that viral mRNA is able to make more capsid proteins. The viral DNA will then replicate. So now you've got a whole bunch of viral DNA, a whole bunch of capsid proteins. Capsid proteins will then surround this newly replicated viral DNA, and when they exit, you now have more virus particles than you started out with up here, right? So this is the reproduction of a virus. So when we come back to this picture right here, and we ask ourselves, what is going on here? What's the purpose? Why are they attaching to this prokaryote? Well, they're attaching to it because they cannot reproduce by themselves. We call them obligate internal parasites. They cannot reproduce by themselves. They are obligate internal parasites. Okay? So basically, you get a cell that's full of viruses. Okay? And they're going to lyse the cell. They will force the cell to lyse. It will just open up. We call this the lytic cycle. Now remember, to lyse means to cut. Remember, glycolysis. Glyco is sweet, lyse is to cut. So this is the lytic cycle. And it literally destroys the host cell like you would imagine that it would. Right? You have your cell, your DNA's been shut down. Now that cell, your cell, has made all of these new viruses. There's so many of them in there that your cell just opens up. Right? It produces so many new virus particles, these what we call phages. And what do you think that those virus particles are going to do now? Yeah, they're going to go infect other cells, right? So if a phage is only capable of reproducing via the lytic cycle, we call that a virulent phage. This phage is a virus. So a virulent phage. It can only reproduce through the lytic cycle. Okay? So now, restriction enzymes have the ability to cut DNA at certain sequences. And some bacteria can use these enzymes as a defense mechanism against viral DNA. Okay? So the viral DNA gets in, and some bacteria can actually start cutting up that viral DNA, just like we do when we do things like when we cut specific segments of DNA with restriction enzymes. It's the exact same restriction enzymes. When you have probably in your class have performed electrophoresis, where do you think that those restriction enzymes came from that you used? The majority of them, what we're using, come from prokaryotes. Then that's exactly in the natural environment. That's what those restriction enzymes are actually for, to start cutting up DNA that is foreign. Okay? So let's just look at the lytic cycle again, just kind of in slow-mo. Okay? So this slide and the three more that's going to come after this all show a virus attacking a prokaryote, okay? And then having that prokaryote make more viruses. Okay? So how do you know this is a bacterium, first of all? Well, we see that it has a single circular chromosome. Right? Don't see any organelles here. There's no membrane-bound organelles, no nucleus, just a single circular chromosome. So we know this is a prokaryote. Okay? So what do you think is going to happen next? Okay? The phage is able to attach to receptors on the outside of this bacterium, which allows it to dock to that cell. Okay? So it attaches to it. It has receptors, and it's able to just attach to that. Then it's going to send its DNA into the host cell, and it's going to degrade the host cell DNA. Okay? So then, just like in a couple of slides ago, we saw that the cell is then used, the machinery in the cell is used to undergo transcription and translation and make more viruses. DNA synthesis of that viral DNA occurs, and transcription and translation occur to make more proteins. Then you have the assembly of, you're going to wrap those newly formed proteins around those newly formed viral DNA, and then there's so many of them that they just burst out. They cause the lyse, they cause the cell to lyse. Okay? So if we look over here at phage assembly, we can see with this bacteriophage, the one that kind of looks like a spider, right? In the head of this thing, we see the viral DNA, right? We see the tail, some tail fibers, right? So all of what you see here in gray or purple, however it looks on your screen, that's protein. The blue is what is the genetic material. And again, that can be DNA, that can be RNA. Okay? So look at the following. What I want you to do is I just want you to look at this following sentence. And I just want you to get with somebody and explain what this means. You need to be able to look at this sentence and explain what this means. So let's just look at it. Viruses replicate via a component assembly model, allowing one virus to produce many progeny simultaneously via the lytic cycle. Okay? So that's what we've been talking about, this reproduction of virus via the lytic cycle. So it is imperative that you be able to explain how that occurs to somebody else. Okay? Now, lytic cycle is not the only thing. It's not the only way that viral DNA can replicate. Okay? The viral DNA can also just lie dormant. Okay? Basically, the phages can reproduce by making copies of its genetic material without actually making any new viruses. Not yet. So if it starts making new viruses, that's the lytic cycle. This is the lysogenic cycle. The lysogenic cycle. What happens is the viral genetic material gets incorporated into the host cell's genome and then stays there. It doesn't shut down the host cell's DNA. It just incorporates itself within the chromosome that is already in the host cell, and then it just stays there. Okay? We call that viral DNA that's now stuck in the host cell, we call that a prophage. Okay? Prophage. Now, think about it. Every time that host cell undergoes binary fission, if we're talking about prokaryotes, or in eukaryotes, every time that cell undergoes mitosis or meiosis, the prophage is copied. So here we look at the difference between the lytic and the lysogenic cycle. Okay? So the phage injects the host cell with its DNA, and it's either going to do one of two things. Either going to go through the lytic cycle, which we've talked about, or it's going to undergo the lysogenic cycle. Now in the lytic cycle, you have the virulent or temperate phage, and in the prophage, it's just the temperate phage only, right? It's just going to stay there. It's just a prophage. In the lytic cycle, we have the destruction of the host DNA. You do not see that with the lysogenic cycle. The lytic cycle, we have production of new viruses, new phages. You don't see that with the lysogenic cycle. Okay? In the lytic cycle, the lysis of host cell causes a release of progeny. Again, in the lysogenic, since we don't have any new progeny, that's not going to occur. Okay, the genome in the lysogenic cycle is integrated into bacterial chromosome as a prophage, which one of two, or two things, is replicated and passed on to daughter cells, and it can be induced to leave the chromosome and initiate the lytic cycle. So let's think about this. We have this prophage that's just sitting there, undergoing. This is a bacterium. It's a prokaryote, so this is just going to undergo binary fission. So every time this thing replicates, it's going to go 1 to 2 to 4 to 8, 16, 32, 64. It's just going to keep that exponential growth that you've experienced with prokaryotes, right? So imagine that this happens, that a phage DNA gets incorporated into the host cell. And then it just starts dividing. Let's say that you have 1,000, 2,000, 4,000, 8,000, and then all of a sudden something occurs to force every single one of those cells to switch over and undergo the lytic cycle. Now think about that. Every single one of those cells, if they undergo the lytic cycle, is now making new viruses. So you can go from happy to sad real quick. So what implications does this have? Okay, if we look at the following four things, how could the following aspects of the lytic and lysogenic cycles increase the rates of evolution? Okay, let's look at each one of these and let's just talk about this. Viruses, let's look at number one. Viruses have highly efficient replicative capabilities that allow for rapid evolution and acquisition of new phenotypes. So how is that going to increase evolution? Well, think about what evolution is. Evolution is the change in gene frequencies in a population over time. Therefore, the faster you get from one generation to the next, the faster the gene frequencies can change, i.e., the faster the evolution of that population. Does that make sense? Let's look at number two. Virus replication allows for mutations to occur through the usual host pathways. So how is that going to increase rates of evolution? Well, mutations are the vehicle by which natural selection can occur. So when you couple this with the rapid reproductive rates the rapid reproductive rates of these host cells, or of these viruses, that's going to increase rates of evolution. Let's look at number three. RNA viruses lack replication error checking mechanisms and thus have higher rates of evolution. So this increases the mutation rate. Thus, you are increasing the effects of numbers 1 and 2, right? Let's look at the final one. Related viruses can combine or recombine information if they infect the same cell. Well, this just increases the diversity of the genetic information of the viruses, thus changing the gene frequencies, right, in that population, thus increasing the evolution. Here is, and if you want to just stop this screencast right here, you can. And again, you can stop this, rewind, fast forward, whatever fits your study method the best. But this is just a really good slide to just kind of look at the difference between the lytic cycle and the lysogenic cycle and understand that just because you have prophages, just because you have cells that are undergoing the lysogenic cycle, that doesn't mean that it can't then undergo the lytic cycle as well. Okay? Now, kind of a new twist. Let's look at some viral reproduction that includes a membranous envelope. So now, what you see here is now we're doing the exact same thing as we did before. Okay? The capsid and the viral genome enter the cell. Okay? Here comes the viral genome. It's RNA. Makes a template. Some of that is used to make glycoproteins. Some of that is used to make capsid proteins. Right? Some of it is used to make copies of the genome, that RNA. So then it encapsulates, just like it did before, in these capsids. Okay? But then when it goes out, it doesn't break out like typically in the lytic cycle. Look what happens. We made some glycoproteins. So these glycoproteins are put on the outside of this this little pocket, this little vesicle that's being made here, right? And so when this virus comes out, it's using, if this is our cell, for instance, it's using our plasma membrane to encapsulate the capsids, right? And then it's putting our glycoproteins, okay, around that. So let's think about this new virus, right? It's got a plasma membrane, just like the host cell does. It's got glycoproteins, just like the host cell does. And so if it comes and docks with another host cell, the host cell, just think that it's another cell just like itself. It doesn't recognize it as an invader, right? And so that virus is protected. It's incognito, right? This allows the virus to go undetected as the host immune system recognizes it as one of its own, and provides receptors on the outside, like we just said, that fit perfectly with other cells. So it's hidden. So let's talk about viruses that attack specifically animals and specifically plants. Okay, so viruses that infect animals have the widest range of RNA genomes, kind of like HIV. Okay? And some of these are called retroviruses. And retroviruses are called retro because they're going to be able to use an enzyme called reverse transcriptase to copy their RNA into DNA. Now let's stop here just for a second. The central dogma of biology, what we've always been taught, is DNA is used to make RNA, and RNA is used to make protein, and you cannot, under any circumstances, go backwards. Unless you have reverse transcriptase. So let's dissect this word, okay? Reverse. We're going backwards, aren't we? Transcriptase. Well, it ends in ase. We know it's an enzyme, right? Transcriptase. So we're probably going to be doing transcription, what? Backwards. Well, what is transcription? Transcription is using DNA to make RNA. So we're going to use that enzyme, reverse transcriptase, to use RNA to go backwards, to make DNA, right? So this is exactly what HIV does. Okay? It is a well-studied system where the rapid evolution of a virus within the host cell contributes to the pathogenicity of viral infection. Okay? So here we see a T4 cell. This is an immune system cell that is infected with HIV. And again, you see how small the HIV is in comparison to that T4 cell? Okay? So let's take everything that we've learned so far. Okay? And let's look at HIV. Okay? It's got the glycoproteins. It's surrounded by this viral envelope by a plasma membrane there. Okay? It's made up of RNA, so it's not DNA, so it's RNA. Okay? Two identical strands. And then these little green spheres here, that's the enzyme. That's the reverse transcriptase. So it comes with that enzyme. So it docks, it injects itself. The actual virus part is injected. Okay, you see the capsid proteins here, you see the RNA, you see the reverse transcriptase. So the viral RNA is right here. Okay, reverse transcriptase is able to make viral DNA. Then that viral DNA will go in, right? Transcription, translation occurs. Okay, here's your new capsid proteins, here's your new mRNA, here's your glycoproteins. And out comes the new virus. Okay? That's how HIV works. Okay? So let's look at plant viruses just for a second. Okay? Most plant viruses have an RNA genome. Okay? And they're going to spread disease in two ways. Now just think, yes, plants get viruses. How do you think that these viruses are spread from one plant to the next? Well, horizontal transmission or vertical transmission. Either horizontal transmission, which means that it enters through the cell walls. Okay, so the viruses are actually attacking those cells, just like what we've been talking about. Or vertical transmission is you pass on from parent to offspring. You pass on that virus. Now, viroids are not actually proteins, okay? But they are just small circular RNA molecules that can replicate within a host cell, but they don't encode any proteins, okay? And they cause a tremendous amount of disruptions in things like regulatory systems that control plant growth, okay? So it's not a fully functional virus, but it can go in there and disrupt a bunch of critical processes. Now, prions are infectious proteins. I just want you to marinate. Let that marinate in your mind for a little bit. Okay? It's not necessarily a virus. It's not a viroid. It's not something that's actually alive. It's literally just a protein, okay, that causes brain diseases in mammals, like Creutzfeldt-Jakob disease in humans, mad cow disease in obviously cows, and then scrappy in sheep. Okay, so how do these proteins reproduce without transcription and translation? Right? They reproduce. They literally turn healthy proteins into prions. They literally turn healthy proteins into prions, and they cause these horrible, debilitating diseases like Creutzfeldt-Jakob disease. Okay, these prions get in there and they start turning proteins into more prions. And as those prions build up, the brain starts to slowly shrink and fill with these holes, which makes this brain kind of spongy. Okay, well, after a while, you're not able to use your brain effectively anymore. Okay, and it's always fatal. It's always fatal.